Hello, College Biology. This is Osmosis Part II, where we discuss examples of osmoregulation. Let's start right away with plant cells. One adaptation that plant cells have regarding osmoregulation is the cell wall. When in a hypotonic solution, meaning that there is a lower solute concentration outside of the cell, meaning there's a higher water concentration outside of the cell, which means there's a lower water concentration inside the cell, so water will move into the cell. When that is the circumstance, animal cells can just fill up with water to the point where they burst, they reach lysis. But plant cells, as you can see here, they have this sturdy cell wall. So the cell membrane will continue to expand as it takes in water from the hypotonic environment, but then when it gets to the cell wall, it stops. The cell wall prevents the expansion. This prevents the plant cell from reaching lysis. So that's one way that lysis is prevented via osmoregulation. So one example of that. Another example is in single-celled organisms. These single-celled organisms have what is called contractile vacuoles. And when they are in a hypotonic environment, water is continually seeping in through aquaporin all around their membrane. So we've got aquaporin all around here. And that water is collected through tubes and tunnels into a contractile vacuole. And we can't see this here because it's a still, but the contractile vacuole will expand, and it's got a little nozzle, a little pore, and it will squeeze all that water back out into the environment. So it seeps into the cell through aquaporin because there's a higher concentration of water outside the cell and a lower concentration of water inside the cell. So it seeps in, then it's collected by the contractile vacuole, and then it's pushed back out when the contractile vacuole squeezes. So you can find videos of this if you want to see that in action online. That is our this of course is also going to prevent lysis, because otherwise the water would build up in the single-celled organism. The next example is sweat, and sweat is a strategy. There's a lot of benefits to sweat, but one thing that sweat does is it helps maintain an environment by removing solutes. So all of these cells are creating waste products, and they are pushing them out into the space between cells. Well, those can get collected by sweat glands, and then the sweat glands will push that waste out onto the skin. Now, if this did not happen, there would be building up of waste solutes between cells, which means over time that space would become hypertonic, and if it were hypertonic, it would then cause the cells to dehydrate and die. So our sweat prevents dehydration when the space between cells is hypertonic. And finally, our fourth example, and there are many, many examples of osmoregulation in the biological world, so this is not the limit, this is just four. We've got kidneys. Cell waste doesn't always go into sweat glands. Cell waste can find itself in our bloodstream as well. And if our bloodstream continues to build the cell waste, it becomes hypertonic. And if it's hypertonic, then as it passes by cells, it will dehydrate those cells, because water will go from the higher concentration in the cells into the blood. So we need to get rid of those solutes to try to maintain an isotonic blood level. That is what our kidneys do. They filter the blood so that it is no longer hypertonic, and they create urine, and then we can pass those waste solutes as liquid waste, preventing dehydration in our cells. So let's review. Some examples of osmoregulation include plant cell walls. So when the plant is in a hypotonic solution, the cell membrane will expand as water enters the cell, and that will continue until the membrane hits the sturdy cell wall, and that will prevent lysis. Contractile vacuoles are features of single-celled organisms. When that organism is in a hypotonic solution, water enters the organism, the vacuoles collect the water, and then they contract, squeezing it out through pores, again preventing lysis of that cell. In sweat, cells deposit their cell waste in the space between cells. They get it out of themselves. But that space becomes hypertonic, which will then lead to dehydration if it's not addressed. The sweat glands will collect the solution and deposit it onto the skin, and that will indeed prevent cell dehydration. And kidneys are working with cell waste that makes its way into the blood. So if a lot of cell waste gets to the blood, it becomes hypertonic. The kidneys filter the solutes from the blood to make urine, and then get rid of it, and that will prevent dehydration. So I hope this helps. struggle well.